Hi, welcome back. Let's continue with our lectures. In the first 3 units, we learned about the hierarchical vocabularies of the gene ontology and KEGG pathway database, and how they are associated with genes and gene products. In this unit, let's look at how you can use them to study your genes and your set of genes of interest. I will use the identification of pathways as an example, but the identification of gene ontology functional categories follows similar ideas and methods. So, you have got a set of genes or proteins from your experiment. For example, a set of 300 upregulated genes. When you compared the RNA-seq data of a type of tumor tissues against normal control tissues. The beauty of the hierarchical structure in the KEGG pathway database and the gene ontology is that it allows you to look beyond the individual genes, to take a bird's eye view and discover higher level patterns such as pathways and functional categories involved in the tumor. But how can you find out which pathways each of the genes belong to? 300 genes are too many to search the KEGG pathway databases by hand one by one. This kind of repetitive tasks need the help of computers. After you map the 300 genes on your list to pathways, you are going to find that the genes are involved in dozens of different pathways, most of which are irrelevant to the biological process in tumor. Most gene sets from most experiments are noisy. So how can you separate out the signal from the noise and find out which were the most significant pathways? When I first joined the faculty at the School of Life Sciences at Peking University in 2004, within the first 3 months, 3 different experimental biologists came to me for help with solving the same problems on this slide, even though they were working on completely different biological problems in different organisms. After searching with the first 50 genes one by one, by hand, their students got tired of it. The situation was worst for one of the biologists who was working on a poorly annotated organism, so there was little existing pathway data at all. Furthermore, the pathways that did come up seemed all over the place and very confusing. They were left wondering which was the real pathway that they should go after in the lab. When 3 good experimental biologists come to you with the same questions, you know that you have got some real questions to work on. Before I tell you what we did to tackle this problem, please pause for a minute to think about how you would deal with these questions yourself. My first instinct was to look for existing tools to do this, but back in 2004, no such tools existed. So we decided to develop one ourselves, which we named COBUS. We published the COBUS software in 2005, which was the first software for identification of significant pathways. Most of the work was done by 3 of my talented former graduate students, Xizeng Mao, Jianmin Wu, and Chen Xie. Xizeng is currently a postdoc at the University of Georgia, Athens in the US. Jianmin is now the head of cancer bioinformatics at Garvan Institute of Medical Research in Australia, and Chen is going to University of Chicago to start his postdoc. The first version of COBUS mapped genes to the KEGG pathways using the KEGG ontology as a controlled vocabulary. To maximize computational efficiency, we first created a relational database to store the data from KEGG. By the way, if you are new to relational databases, you can watch a supplementary learning video that Dr. Chen Xie in my group had made for you. We wrote a Python program to parse all the KO entries in KEGG and stored the data into the relational database, creating a table for the KOs, a table for the pathways, and a table linking the KOs to the pathways. Then, we parsed all the gene entries in KEGG and again stored the data into the relational database, creating a table for the genes, a table linking the genes to the pathways, and a table linking the genes to KOs. Now, given the gene that you're interested in, how can you find out which pathways it is involved in? If your gene is a known gene in a well-annotated species such as human, mouse, and other model organisms, it is likely that the gene has already been annotated in KEGG. So you can use simple ID mapping. You provide the ID of the gene in the format of GenBank GI number, or Untrace gene ID, or Ensemble gene ID, or UniProt KBAC, and Cobus looks into its relational database to retrieve the pathway linked to the gene. If your gene is a newly discovered one, or is from a poorly annotated non-model organism species, then ID mapping wouldn't help you much. To solve this problem, we implemented sequence mapping based on the assumption that homologous genes in different species are likely to be involved in the same pathways. You provide the sequence of your gene of interest, the query sequence. Cobus has already parsed the genes in KEGG into its relational database, including the sequences of the KEGG gene. Cobus then ran BLAST to find the KEGG genes that have similar sequences as your query. Here, a default e-value cutoff of 10 to the power of -5 is applied. For some reason, not all the genes in KEGG have been linked to pathways. So we applied a default rank cutoff of 5, meaning that if all top 5 BLAST hits do not have a pathway linked to it, we give up. It could just be that this particular gene family has not been well studied. It might be better to return no result here than to return too many false positive results. Of course, in your own analysis, you may want to relax the cutoff or make it even more stringent. You can do this by easily adjusting the parameters in Cobus. Most genes in KEGG are linked to KOs, and the KOs are linked to pathways. By following the links from your query sequence to its BLAST hits in the KEGG genes to KOs to pathways, you can computationally annotate your query genes with its pathways. How can you evaluate the accuracy of this kind of pathway annotation by sequence similarity? We defined 2 parameters. The first is precision. You may remember from the previous week's lectures that precision is defined as the number of true positives divided by the sum of the number of true positives and false positives. It measures among all the genes annotated a pathway by the sequence mapping method what percentage of the annotation is correct. You can imagine that if you use very stringent e-value and rank cutoff, then your precision can be quite high. However, the downside is that you can only make annotations for a small number of genes. So, in addition to evaluating precision, you also want to evaluate coverage. The coverage is defined as the percentage of correct pathway annotation among all query genes, no matter if the query gene got an annotation or not. So there is a trade-off between precision and coverage. Genes in the yeast genome was pretty well annotated in KEGG. So we removed all yeast genes from the KEGG datasets, ran Cobus on all the yeast genes, and then compared the computational annotations with the original annotations in KEGG. This figure shows the precision in the y-axis and the coverage in the x-axis when different cutoffs of e-value and rank were applied. Overall, the performance of Cobus annotation seems good, with over 89% coverage and 93% precision in the worst case. Our default e-value cutoff, 10 to the power of -5, and run cutoff 5 have coverage of 90.3% and precision of 94.4% on the yeast dataset. In addition, Scopus was able to provide annotations to a few yeast genes that were not previously annotated in KEGG. Now, after you generate pathway annotations for your set of genes, either by ID mapping or by sequence mapping, you will often find that dozens of pathways show up. Which pathways were the most significant ones? The most obvious data is to look for pathways that covers the most number of genes in your input gene set. This is an intuitive idea, but unfortunately it suffers from the fact that some pathways are intrinsically much larger than others. So purely by chance, these pathways may cover more genes in your input set. It is important to remember that any gene set can be noisy and may contain genes that are noise, meaning that they are irrelevant to the biological process you study. In the worst-case scenario, the whole gene set may just be a random collection of genes. In that case, you want the software to return no pathways. You don't want the software to give you a false positive pathway and lead you to spend lots of time and effort towards a dead end. So, the software needs to have a way to separate truly relevant pathways from random chance. One way to do this is by identifying statistically enriched pathways. Let's see how it is done in Scopus. In Scopus, each pathway is tested one by one. For a specific pathway, let big N be the total number of genes. For example, all the genes in the whole genome or all the genes with probes on the gene expression microarray. This is your whole universe of genes, often called the background. Here we only include genes mapped to pathways and ignore genes that are not mapped to any pathway. Let big M be the number of genes in this particular pathway. As we had discussed at length so far, this information is available in KEGG. Now, you get a set of genes from your previous experiment that you want to analyze. Let's call them query genes. The query genes must be a subset of all the background genes. Let small n be the total number of query genes. They are often called the foreground. Some of these query genes will fall into this particular pathway. Let small m be the number of query genes that fall in this particular pathway. Now, out of all big N genes in the genome, big M genes belong to this pathway. Out of all small n genes from your previous experiment, small m genes belong to this pathway. Is this pathway special for your experiment? How do we define special? In statistics, this question is often looked at from the opposite angle. When we take n genes, small n genes, from all big N background genes, what is the probability of getting small m genes from a specific pathway of size big M just by chance? If this happens just by chance, then this pathway is not special for your experiment. The just by chance possibility is called null hypothesis. The p-value, which is used everywhere in statistics, is the probability that the data have occurred just by chance, assuming that the null hypothesis is true. If the p-value is very small, say less than 1 in 100 or less than 1 in 20, then your observation is unlikely to have occurred just by chance. Then it is likely that this particular pathway is special for your experiment. You reject the null hypothesis. The smaller the p-value, the better. So now the question becomes how we can calculate the p-value. Depending on the nature of the data and the null hypothesis, different statistic tests have different ways of calculating the p-value, assuming different probability distributions. Let's focus on this particular case here. Here, the p-value is more precisely defined as the probability of observing data at least as extreme as this, assuming that the null hypothesis is true. Again, the null hypothesis means that there is no real signal in your gene set. The gene set you got from your previous experiments only happened by random chance. So when you randomly draw n genes from a total set of big N genes, what is the probability that I of the genes fall in a particular pathway of size big M? This is described by the hypergeometric distribution to be this formula over here. So the p-value in this formula, so the p-value is this formula summed up for I from small m to big M. And it is equal to 1 minus this formula summed up for I from 0 to small m minus 1. And we get a p-value. If the p-value is less than or equal to 0.01, then the pathway is considered unlikely to have occurred by chance. And you have a positive discovery, although the discovery has a 1 in 100 chance to be a false positive. Often the p-value cutoff is relaxed to 0.05. You have a better chance of getting a positive discovery, but your discovery has higher false positive rate, now at 1 in 20. So far, we've been talking about testing one pathway. To find the significant pathways in your gene set, you usually need to test all hundreds of pathways. Every test has a false positive rate. I hope you still remember this table. When you test multiple hypotheses, your chance of getting false positive discoveries accumulates. Some people test multiple hypotheses but only report the one significant finding. This practice is wrong and dangerous, as such result tends to have low reproducibility. When you are a student, you may think that it is bad to have no positive result and no paper to publish. But the truth is that it is even worse to publish a false positive result that cannot be reproduced. Good statistics keeps us honest. One way to estimate your overall false positive rate is to calculate the family-wise error rate, or FWER, defined as the probability of getting one or more false positives among all the hypothesis tests. This is very conservative and often considered too conservative. A good alternative, which is widely used, is to calculate to control the overall false discovery rate, or FDR, defined as the expectation of false positive over true positive plus false positive. This is much less conservative. Cobas uses FDR correction for multiple hypothesis testing. Cobas has a web server interface that is pretty easy to use, accessible from the URL down here. We will provide a supplementary video of a student presentation that will show a demo of how to use Cobas. So I will only briefly mention the major steps here. First of all, version 2.0 of Cobas tries to provide one-stop shopping. It incorporated data from not only KEGG, but also PID, BioCarta, Reactome, BioSeq, Panther, and Gene Ontology. It also incorporated data from several gene disease association databases, such as OMIM, KEGG disease, FONDU, GAD, and NHGRI GWAS catalog. So you can find out which diseases are most relevant among your set of query genes. Qubits has two steps. The first step annotates the input genes with pathways, either by ID mapping or by sequence mapping. The result of annotate is shown here. It can be easily used as an input to the second step, identify. For both annotate and identify steps, you can either use the default settings or adjust many of the parameters. The final result is a list of pathways ranked by their relevance to your set of genes. Both the p-value and the FDR-corrected p-value are shown. Pathways with FDR-corrected p-value less than or equal to 0.05 are considered significant. You can also download and run a stand-alone version of Qubits instead of using it online. Again, there are a bunch of parameters that you can set. Once you learn to work in the Linux environment, you'll find that it is often more convenient to work with standalone versions of software than with the web version. Qubits is only one of the pathway identification methods. It belongs to the group of overrepresentation analysis methods. Two other groups of methods include functional class scoring and pathway topology. For example, Qubits David and PAGED belong to the first group. GSEA is based on functional class scoring, and SPIA is based on pathway topology. If you're interested in the problem of pathway identification, I encourage you to learn about these other methods as well. In the next unit, I'll show you how you can apply the concepts and methods you learned in this week's lecture so far to study a real problem and discover very interesting patterns. I look forward to seeing you then.